Episode 5 of the BioLogic podcast. Today I'll be discussing a very important class of molecules called carbohydrates. You might know carbohydrates by their more common name, sugars. I'm going to hit you with a little technical terminology here, so try and follow me. Carbohydrates are sugars that can group together to make polymer chains. Sugar monomers consist of a carbon backbone with a carbonyl group and numerous hydroxyl groups. A carbonyl group is a carbon atom double bound to an oxygen atom. A hydroxyl group is an oxygen atom bound to a hydrogen atom on one side and a carbon atom on the other. Carbonyls can be on the ends or somewhere within a chain. Carbonyls on the end of a chain are called aldoses, while those within a chain are called ketoses. Because carbohydrates are just chains of carbon atoms with a carbonyl group and a few hydroxyl groups, different carbohydrates may have the same chemical formula. If you think about a hydroxyl group, its formula is CH2O. The oxygen and carbon are both bonded to a hydrogen atom. With the exception of the carbonyl, whose chemical formula is CHO, the rest of the polymer is basically composed of repeating CH2O groups. The carbon in the backbone shares a bond with a hydrogen atom, and the oxygen in the hydroxyl group also shares a bond with a hydrogen atom. This means that all carbohydrates with, for example, 6 carbons in the backbone will share the same chemical formula, CHO for the carbonyl group, and CH2O(6) for the rest of the molecule. This is problematic because the chemical formula doesn't tell you everything you need to know to identify the specific species of Hexose carbohydrate. Besides the length of the chain itself, the features that differentiate carbohydrate molecules from one another are the placement of the carbonyl group in the chain and the orientation of the reactive hydroxyl groups along the chain. Hydroxyl groups can be attached to any carbon in the chain. They can be oriented in alpha or beta positions, and they can share the carbon chain with a number of other hydroxyl groups. This wide variety in structure gives each carbohydrate a unique function and reactive capacity. Just like the proteins I covered in episode 3 and the nucleic acids I covered in episode 4, carbohydrates are polymers. Polymers, as I've said before, are long chains or structures composed of repeating subunits called monomers. The monomers of the carbohydrate class are called monosaccharides, mono meaning one, and saccharide meaning sugar. There are a huge variety of monomers, which can be combined in a huge number of ways to create a variety of different complex carbohydrate molecules. 2 monomers bound together make a disaccharide. When a small group of 3 to around 10 or 12 monosaccharides binds together, they're called an oligosaccharide, where oligo means few. Think of an oligopoly, which is a government ruled by a few privileged people. In much the same way, an oligosaccharide is composed of just a few saccharides. Larger carbohydrate polymers are called polysaccharides. Polysaccharides can be composed of a single repeating monomer or a repeating series of different monomers. The monosaccharides bind together with glycosidic linkages, covalent bonds formed through a condensation reaction. Because they all form covalent bonds through condensation reactions, glycosidic linkages are comparable to the peptide bonds between amino acids and proteins, and the phosphodiester linkages between nucleotides and nucleic acids. Similarly, all of these bonds can be broken through hydrolysis reactions. Sugars can be organized according to the number of carbons in their polymer chain, given a numerical prefix attached to the ose suffix. A sugar with 3 carbons is called a triose, 4 carbons and it's a tetrose. 5 carbons makes it a pentose, like the sugar molecule in the backbone of nucleic acids. The ribose sugar in DNA and RNA is a pentose sugar. This naming convention continues logically. 6 carbons makes a hexose, 7 makes a heptose. Recall how I numbered the carbons in the nucleic acids as a tool for organization and efficient communication. Carbohydrates can be numbered in a similar way. To conceptualize the carbohydrate molecule, imagine a zigzagging line. Each point on this zigzagging line, that is, each point connecting 2 lines, is a carbon atom. Each line, therefore, represents the bonds between the atoms. In simple carbohydrates, the carbon atoms naturally arrange in this linear zigzagging structure. The numbering starts at the end closest to the carbonyl group. So let's say the carbonyl group is an aldose on the left end of the chain. The carbonyl carbon, the farthest left atom, is labeled the 1' carbon. The rest of the carbon atoms in the chain are labeled sequentially going from left to right. Let's make this a little more complex. What if the carbonyl group is a ketose somewhere within the chain but near the right side? In this case, the 1' carbon is the carbon atom on the far right end of the chain, as this end is nearest the ketose. The numbering would then go sequentially, right to left. In nature, it's really uncommon to see linear structures in carbohydrates longer than 5 or 6 carbons. When the backbone gets any longer, it tends to form ring structures. The ring is formed when the carbonyl carbon and a carbon with a hydroxyl group bind together. The oxygen atom in the carbonyl group is electronegative, so by induction it pulls the shared electrons closer to itself. Furthermore, the resonance structure of the carbonyl oscillates between two states, with a double bond between neutral atoms and with a single bond between a positively charged carbon atom and a negatively charged oxygen atom. These qualities make the carbonyl carbon partially positively charged and able to attract the electronegative oxygen atom in the hydroxyl group. Through a reduction reaction, the carbonyl is turned into a hydroxyl group, while the oxygen atom in the original hydroxyl group becomes an ester between its original carbon and the carbonyl carbon. This is how a linear carbohydrate molecule interacts with itself to form a ring structure. It's important to understand this reaction is spontaneous, and it doesn't involve the loss of any atoms. Many sugar molecules have numerous hydroxyl groups that they could hypothetically bind to, but the spontaneous reaction that they actually undergo will be the one that requires the least energy. The reaction that requires the least energy will be the one that breaks as few covalent bonds and moves as few atoms around as possible. Consider the hexose sugar, glucose. It possesses a carbonyl on the 1' carbon and 5 hydroxyl groups. In a water solution, glucose spontaneously forms a ring structure connected through its 1' and 5' carbons. As glucose is a hexose sugar, it has 6 carbons. The 6th carbon juts up and out of the ring, like the 6th carbon in the ribose sugar of a nucleic acid. All of the hydroxyl groups attached to the linear molecule become oriented in one of two positions when the molecule takes on a ring structure, called alpha and beta positions. The alpha and beta positions are determined by a hydroxyl group's orientation relative to the plane of the ring. The top side of the plane is the side that has the 6th carbon atom jutting out of it. A hydroxyl group in the beta position will be oriented such that it also juts out above the plane. A hydroxyl group in the alpha position, by contrast, will be oriented below the plane of the ring. As hydroxyl groups are the reactive groups on carbohydrates, involved in glycosidic linkages and other interactions, the orientation and geometry of the hydroxyl groups influences how the sugar interacts with other molecules. Glucose can take two forms, alpha glucose and beta glucose, depending on the orientation of the hydroxyl group on the 1' carbon. For all glucose molecules, the 2' and 4' carbon atoms have hydroxyl groups in the alpha position, and a hydroxyl group in the beta position on the 3' carbon. Alpha glucose has the 1' hydroxyl group in the alpha position, while beta glucose has the 1' hydroxyl group in the beta position. When two glucose molecules bind together, they form a disaccharide called maltose. The two glucose molecules within a maltose molecule share an alpha-1,4 glycosidic linkage. This signifies the type of bond between the 1' and 4' carbons. In this type of bond, the oxygen atom connecting the monosaccharides sits above the plane of the ring, whereas in beta bonds, the oxygen atom is positioned above the plane of the rings. Virtually all monosaccharides bind together to form polymers through the alpha-1,4 or beta-1,4 glycosidic linkages. Long chains of glucose monomers bound through alpha-1,4 glycosidic linkages will form helix structures. Remember how I said that glycosidic linkages were similar to peptide bonds and phosphodiester linkages? These bonds do share some qualities, but not all of them. Peptide bonds and phosphodiester linkages are binary. They have two ends involved in linkages, which are always in the same spot on each nucleic or amino acid. In carbohydrates, however, hydroxyl groups can be anywhere along the ring, oriented either above or below it. This means that sugars don't form linear molecules like polypeptide chains or nucleic acids. Instead, they form irregular branching structures due to the highly variable nature of the geometry of the hydroxyl groups. It cannot be understated how important the structure of the carbohydrate is for its function. The difference between alpha and beta positions on a particular carbon may seem small, and yet this can be the quality that differentiates structural carbohydrates from energy storage carbohydrates. I'll go into more detail on the functional role of carbohydrates later in the episode. Consider the following kinds of carbohydrates: starch, glycogen, chitin, cellulose, and peptidoglycan. Each of these molecules is hugely abundant on the planet Earth, due to their ubiquity in almost every group of living organisms. Starch is a sugar used in plants to store chemical energy. The starch polymer is built exclusively out of alpha-glucose monosaccharides connected with glycosidic linkages. There are two types of polysaccharides that compose any given quantity of starch: the branching amylopectin and the unbranching amylose. Amylose is a simple linear polysaccharide with alpha-1,4 glycosidic linkages. Amylopectin shares the alpha-1,4 linkage structure with amylose, but it also has branching chains. Every 30 monomers or so, a chain of alpha-1,4 linked monomers will bind to another chain of alpha-1,4 linked monomers, binding through the 1' carbon on one strand and a 6' carbon on the other. Glycogen is a sugar that's similarly used to store chemical energy, but it's present in animals instead of plants. You and I store our glycogen in our liver and in our muscles. During periods of physical exertion, these huge and wildly branching glycogen polymers are broken down into glucose subunits, which are then used for energy. The structure of glycogen is very similar to amylopectin, except the branching is 3 times as dense. Branches form every 10 monomers instead of the 30 monomer intervals in amylopectin. Chitin is a structural polysaccharide found in the harder tissues of numerous animals, including the exoskeletons of insects and crustaceans, the beaks of cephalopods, the scales of fish, and the tongues of mollusks. Chitin is composed of monomers called N-acetylglucosamines, or NAGs, or NAGs, connected through beta-1,4 glycosidic linkages. Because of this linkage structure, every other monomer is flipped upside down. The polymer forms a straight line, not a helix, allowing the polymer strands to be laid next to one another. The chitin polymers form hydrogen bonds between one another, creating a stiff, sheet-like structure. NAG possesses an acetylamine group in the 2' carbon beta position, which forms particularly strong hydrogen bonds with the other acetylamine groups and other monomers. These sheets of chitin are deposited layer upon layer, forming the tough exoskeleton of a beetle or the sugary wall of a fungal cell. Cellulose is also a structural polysaccharide, except it exists in the cell walls of plants and most algae species, not fungus and insects. Like chitin, cellulose monomers are beta-glucose molecules connected with beta-1,4 glycosidic linkages to form long strands. Where chitin monomers have an acetylamine group in the 2' carbon beta position, cellulose monomers only have a hydroxyl group. Anyway, these strands of cellulose polymers are organized parallel to one another, where they form multiple hydrogen bonds and coalesce into a strong sheet or wall. These sheets of cellulose are very regular, as the covalent and hydrogen bonds form a clean, grid-like pattern. Peptidoglycan is the third structural polysaccharide. Bacteria have cell walls, but because cellulose and chitin are difficult, if not impossible, for most bacteria to produce, they use peptidoglycan. Peptidoglycan is a complex polymer. Each monomer is composed of 2 different sugar molecules bound with a beta-1,4 glycosidic linkage. The first sugar is N-acetylglucosamine, the same sugar used in chitin. The second is N-acetylmuramic acid, which is bound to a chain of 3 to 5 amino acids. So N-acetylmuramic acid is part sugar, part protein. The protein segments on one peptidoglycan strand form peptide bonds with the protein segments on other peptidoglycan strands, which function like the hydrogen bonds in other polymers to create sturdy, sheet-like structures. Here's an interesting fact. Bacteria can be categorized based on their cell walls. By using a test called Gram staining, which involves staining with a dye called crystal violet, one can differentiate between gram-positive and gram-negative bacteria. Gram-positive bacteria have thick peptidoglycan walls that will absorb a lot of the dye, making them appear a dark purple. Gram-negative bacteria have thin peptidoglycan walls that absorb little, if any, of the dye, making them appear a pale purple or failing to stain them at all. Consider the fact that virtually every cell in existence uses starch or glycogen to store chemical energy. Consider the fact that every plant on the planet is composed of cells wrapped in a sheet of cellulose. Consider the fact that every insect is encased in a shell of chitin, as are the cells of all fungi. While each cell may be small, when you begin adding up all the billions and billions of cells in each plant or fungus, or the billions of molecules in the exoskeleton of a bug, and then you add up the billions and billions of individual organisms that exist across the Earth, the total mass of each group of carbohydrate molecules is unbelievably huge. Despite their presence and critical function in every modern cell, it's very unlikely that carbohydrates were involved in the genesis of life. Carbohydrates do not contain code-like sequences of repeating structures like a nucleic acid, so they cannot act as a template for their own replication. For that matter, no carbohydrate would be able to initiate its own replication because they are, structurally, too simple to be effective catalysts. The reactive hydroxyl groups can only engage in simple, limited reactions. Proteins and nucleic acid ribozymes, on the other hand, come in very complex shapes that can catalyze all manner of reactions. Perhaps most fundamentally, the glycosidic linkages that connect carbohydrate monomers can't be formed without a particular suite of catalytic proteins. For these reasons, it's unlikely that carbohydrates were involved in the origin of life. Think about this. The very first cells to emerge were far smaller and simpler than the large modern cells that compose complex eukaryotes like you and I. Without the complexity of organelles or specialized functions, the energy demands on these first cells would have been pretty minimal. There was no real need to invest in some kind of energy storage. But then cells started to evolve. Specialized functions or features began to appear, differentiating the early cells and making them slightly larger, more energy-hungry. As the early cells became more and more sophisticated, their need for energy increased. Their membranes grew until they required mechanical support. Eventually, the cells would need to find a way to store energy for later use, because their sophisticated metabolic and functional activities consumed more energy than what was immediately available. These cells would also need to utilize some kind of reinforcing framework to support the growing size of their membranes and the biomolecules they synthesized. Carbohydrates are the class of molecules that stepped in to fill both of these gaps, providing an efficient and effective platform for energy storage, as well as providing structural integrity to the walls of the cell. Sugars are common as structural components in larger biomolecules. Your body, for example, produces amino acids through a series of chemical reactions that starts with a sugar molecule. Half of the subunits in the backbone of your nucleic acids are sugars, ribose sugar in your RNA, deoxyribose in your DNA. But how else are carbohydrates involved in the structure of your biomolecules and your cells? Carbohydrates are durable, elastic molecules that show levels of structural organization kind of, but not exactly, like the primary, secondary, tertiary, and quaternary structure of proteins. Recall how earlier I described polymer strands lining up beside one another and connecting through numerous hydrogen bonds. Groups of these hydrogen-bonded strands form sheets, which can wrap together to form fibers, which themselves are piled and crisscrossed into a giant elastic sheet. Sometimes the main structure is just a massive, strong fiber. Cellulose, for example, forms fibers made out of 70 to 80 polymer strands. The fibers pile and overlap, covering the cell in a flexible but sturdy sheath or sheet. The fibrous sheets are very strong and durable, and they're able to withstand the tension and compression forces that are exerted on a typical cell. Recall that the structural sugars I described earlier all share beta-1,4 glycosidic linkages. The structure of the beta linkages makes the fibers very durable, inaccessible to water molecules, and thus insoluble in water and resistant to being broken down by hydrolysis. This is also why structural carbohydrates are so effective. Consider the cellulose, or dietary fiber, found in plant foods. Fruits and vegetables are high in cellulose, as are whole grain breads with seeds. Cellulose is indigestible, so when you eat it, instead of being digested, it coalesces into a spongy ball that helps moisturize and lubricate your intestines as it moves through them. The wet fiber mass assists in smooth digestion and easy, regular bowel movements. Carbohydrates also play a very important role in cell-to-cell communication, cell signaling, and cell identity. The cell membrane is composed of a bilayer of phospholipids studded with various proteins. I'll go into more detail on lipids and membranes in the next episode, but I need to mention this briefly for now because of the importance of glycoproteins. Glycoproteins are a sugar-protein combination, wherein the protein is studded in the membrane, and the sugar extends off of it, reaching away from the surface of the cell. Glycoproteins possess a huge variety of oligosaccharides, which play a really critical role in the proper functioning of your body. These oligosaccharides are used by your immune system to identify cells that belong to the self, cells that are healthy and belong to your body. Cells that don't have these identifying sugars are perceived by your immune system as not self, or not a part of you, and they become targets for the T cells, natural killer cells, and other antigen-destroying immune cells. These identifying sugars are not the same for every cell in your body, Different types of cells have different groups of identifying sugars on their glycoproteins. Your neurons have a different set of glycoproteins than your dermal cells, which are different than those in your muscle cells, which are different than those in your retinal cells, and so on. For example, a sperm cell and an egg cell will meet, fuse, and become a fertilized egg, which will then grow into a zygote. But in order for the sperm to fuse to the egg and initiate fertilization, it has to find a glycoprotein with a carbohydrate that it recognizes and can bind to, namely a glycoprotein of its own species. There are many reasons why organisms from two different species cannot interbreed, and the ability for sperm to find a recognizable carbohydrate on the egg cell is one of those reasons. A successful binding will initiate the absorption of the sperm cell into the egg and begin the development of a new individual. Besides being used for structural support and cell signaling, carbohydrates are critically important for their role in energy storage. Remember how I said that monomers connected with beta-1,4-glycosidic linkages made for good structural proteins? Well, what about the monomers that are connected with alpha-1,4-glycosidic linkages? These monomers form polymers that are easy to break down with hydrolysis. This is good for a complex molecule meant to store energy. Chunks of it can be readily broken off when needed and metabolized to supply energy to the cell. For alpha-glycosidic linkages in glycogen, an enzyme called phosphorylase is required to initiate hydrolysis. Your muscle cells and liver cells have higher than average quantities of phosphorylase, as these are needed to break down the glycogen stored in those tissues. Understand, however, that virtually all cells have some level of phosphorylase, because all cells need to access their troves of stored chemical energy. This energy is then used to power the various functions of the cell. You might think I'm dancing around the issue. How is energy literally stored in carbohydrate molecules? How does the cell access this energy in a usable way? Well, enough talk then, let's get to the point. Chemical energy exists in the bonds between atoms. Those shared electrons took energy to pair, and it takes energy to break them apart. Similarly, it takes an energy input like heat to break hydrogen bonds, although breaking hydrogen bonds doesn't yield energy like a covalent or ionic bond. Consider the carbonyl double bond, or the hydroxyl single bond, between an oxygen atom and a carbon atom. The carbon is electronegatively neutral, while the oxygen atom is very electronegative. Because of this quality, the oxygen atom holds the shared electrons closer to itself, which reduces the potential energy of the bond. This makes carbon-oxygen bonds bad at energy storage. If you watched episode 2, you might remember a metaphor or analogy that I made for explaining electronegativity with planets and gravity. I think the metaphor could be useful in this situation as well. Imagine some kind of mass, something generally big like a bus without wheels. Imagine pushing this bus without wheels while it sits on flat ground. You can get a few friends to help you, but chances are the bus isn't going anywhere. What if you and the bus were, for the sake of hypothetical argument, floating in a dark expanse of microgravity? Your spacesuit has a thruster pack, but it's kind of weak and can only produce the same amount of thrust that you could on the surface with your meat body, like when you were pushing against the grounded bus. So now in microgravity, you apply the same amount of force to the bus, and in microgravity it will slowly accelerate. If a big purple gas giant suddenly appeared nearby, the bus would be attracted to it and would accelerate as it fell into the gas giant's gravity well. In this way, the bus has more potential energy here than it does sitting on cinder blocks in someone's backyard. Electrons are the same way if you consider the valence shell akin to the surface of the planet. Near the surface, objects have less potential energy. Similarly, electrons held close to a strong source of electronegativity like an oxygen atom have very low potential energy. If oxygen's strong electronegativity is causing the bonds it makes to have low potential energy, what about carbon atoms? Carbon atoms are electronegatively neutral, as are hydrogen atoms. As you probably surmised, this is where the energy is stored in carbohydrates. In a carbon-carbon bond and a carbon-hydrogen bond, the electrons are shared equally between the atoms. Continuing with the gravity metaphor, this is like the potential energy of a large item on the edge of a gravity well. It has large potential energy here, but it's easily removed from the gravity well. I'm sure there are chemists out there itching to tear my metaphor apart, but if it helps you understand the concept in biochemical terms, then that's what counts. Anyways, the alpha-glycosidic linkages of energy storage carbohydrates can be broken to yield metabolically accessible glucose monomers. This is where the phosphorylase and amylase come into play. These enzymes are part of the protein suite that is necessary to conduct hydrolysis on the glycosidic linkages of glycogen and starch, respectively. The glucose monomer is then consumed in a process called cellular respiration, which I'll explore in a future episode, where the glucose molecule's bonds are broken down, the bond energy harvested, and the atoms reconstituted into other molecules. These other molecules include a variety of energy storage carbohydrates, but consist primarily of a nucleotide called ATP, or adenosine triphosphate. ATP is synthesized from several precursor molecules, including molecular oxygen, an inorganic phosphorus atom, the carbohydrate, which is consumed for energy, and a molecule called ADP, adenosine diphosphate. The energy in the chemical bonds of the glucose molecule are used to attach the inorganic phosphorus atom to the other phosphorus atoms in ADP. Your cells then consume ATP to access this energy in usable ways. ATP releases energy by having its phosphorus groups broken off, and the high bond energy used to power another chemical reaction, or really any other process that requires energy. In these ways, carbohydrates can provide both structural support to cells and a format for storing energy on a molecular level. As useful as they are, carbohydrates pale in comparison to lipids when it comes to energy storage. I mentioned lipids earlier in this episode, and I'll discuss them in greater detail in next episode, episode 6. That's about it for this one, though. I hope you learned something neat. Thanks for listening. If you enjoyed the podcast, tell your friends, tell your teachers, tell your classmates. Hit the like button and subscribe so you can see new episodes when they release. The BioLogic podcast is kept alive by the support of listeners like you. If you want to help support the podcast, please consider buying some merchandise in our official store or becoming a patron by visiting our Patreon page. The official BioLogic store can be found at redbubble.com/people/biologic/shop. In the store, you can buy merchandise like t-shirts and coffee mugs and hoodies. All the products have really cool biology-themed artwork on them that I've designed myself. The official BioLogic Patreon page can be found at patreon.com/biologicpodcast. The support of Patreon patrons is very important for the show. Longtime patrons are rewarded with exclusive episodes on fascinating topics and will have their questions prioritized in AMA episodes. Thanks for your support, and as always, thank you for listening.